Hello, Ritika.
Yes, sir, I'm here.
Yes, it's 10:31. I mean, it's 1 minute late already.
All right, so should I start?
Yeah, please. I mean, I thought you were going to start 1 minute ago.
Okay, cool. So, hello and welcome everyone. Thank you for joining us for today's seminar. I am Ritika, an alumnus of IBB Bangalore, and your host for today's webinar from Coronavirus webinar series. The motto of the organizing team is to spread awareness amongst the audience without a formal training in biology. Today's talk, titled as Immune Response to Coronavirus and the Implications of Immunotherapy, will be presented by Dr. Srini Kaveri, the Director of Research at INSERM Paris. He also holds the position of Director at CNRS Bureau in the French Embassy in New Delhi. A veterinarian by training, he received his PhD from the Pasteur Institute and did postdoctoral work at the University of California. His work is related to the molecular mechanisms of immunopathology and immunotherapy. A very warm welcome to you, sir. Thank
Thank
you
you.
for joining us today.
Thank you.
Just a little housekeeping before we get started. If you have any questions during the presentation, please drop them to in the comment section on our YouTube live stream. We will have time for your questions at the end of the talk. Now, without further ado, we will turn the time over to Dr. Kaveri, who will be talking to us about immune response to coronavirus and the implications of immunotherapy. Over to you, sir.
Thank you, Ritika. Thank you, Neeraj. Thanks also to the person who initially got in touch with the Kishan, I guess. And yeah, it's a pleasure being with you this morning,
Thank
although virtually. I am not new to IBAB actually. I do not know whether some of you have already seen me there. I am on your adjunct, and therefore I keep coming there almost every year, at least once a year, and therefore it's for me not new place, and it's just coming to my place. Okay, so thank you very much, IBAB, and thanks to everyone. And before I start, I would also like to thank my colleagues in Paris. As you can see, it's a bright, beautiful morning out in Paris, just like that particular day when I took this picture. That was some time back. This is only a representative picture to say thanks to my colleagues and students. As you can see, we are quite a happy little group. We are about 22 people, and it's quite an international team, as you can also probably see. We have people from different parts of the world, and on my extreme left is Chavdar, one of my oldest collaborators from Bulgaria, and on my extreme right is Vani Janaki Raman. She is now an assistant professor at IIT Madras. And then a whole bunch of people, I will have probably time to thank them during the course of my talk. And as you can see, you are in the Jardin de Luxembourg, like Jardin de Luxembourg, which is right in the heart of Paris, not far from Notre Dame. At the far end there, you see the Senate building. And if you walk on the right side here, I mean, past Wani, you will be seeing the Pantheon, which is one of the most beautiful monuments to commemorate all the great people of France. And if you walk past this, just about 300 meters or so, you will be on Boulevard Saint-Michel, Boulevard Saint-Germain, and at that crossing is our lab. And sometimes when they're in mood, these people are also in the lab actually. And when they're in the lab, they're very, very efficient. Uh, I just would like to thank all my Indian students and colleagues, because I have had long, long association with India, and therefore I have hosted quite a few students in the lab as either PhD students or postdocs in the lab, and I thank them all too. Um, since many of the questions that we ask in the lab are clinically oriented, I would like to thank all my clinical colleagues and collaborators. On the left is all those people in Paris and France. And on the right, you can see people from different parts of the world. This is only a representation. I mean, I cannot give the whole list of all my collaborators. That would be too big. Just a few of them. And I would like to point out people from the Indian Institute of Science, Nagraj, Balaji, D.N. Rao, and from Pondicherry, Jipmer, and from Bombay and other places. So before I start, I have a small disclaimer. I'm talking to IBAB students, I guess, and I am talking to biotechnologists or the future biotechnologists and bioinformaticians, that too of Bangalore, so the hotspot for everything on all these domains. So I'm actually feeling a little guilty because in the last, I guess, 6 months or so, there is so much information on coronavirus that what I'm going to say is probably not new to you, but I still go ahead and I present my views on coronavirus, particularly from an immunologist point of view. Okay, this is purely from an immunologist point of view. Because these days, with the social media being so strong, everyone seems to be an expert, and there are more experts than the very cases these days. Well, let me start off. As I said, I'll focus mostly on the immune response, and as I have put in the abstract, most of the damage that happens following COVID-19 is also due to an abnormal or uncontrolled immune response. And therefore, in my opinion, it is very important to understand the very basics of what happens in immune response. And therefore, let's start off with that. And before that, I would like to share with you a secret and probably a sacred mantra. This you can recite almost every morning as a biology student. To me, this mantra is how our well-being, our health / disease, if you're not in good health, you have some discomfort, which means disease, is controlled by 3 very, very important factors. The genes. We all know how important are genes, how important is our genetic makeup, our heredity, and all that we have inherited from our folks. Because those genes control every function in the body. Those are the physiological functions, the second ring that I have shown on the left. Every function, as you all know, is regulated by genes. But equally important, rather even sometimes appears more important, is our environment, which can tilt, although it cannot probably directly shape the genes, it can tilt a lot in terms of health and disease. And therefore, it is very important also to study and to have an idea of what this environmental influence is on our health or disease. When I talk of environment, I just don't mean a beautiful garden out there where the hero and heroine are singing. I don't mean only that. I don't mean only the toxins in the air or food or water. I also mean the noise pollution, the ultraviolet rays, the weather, the climate, the urban design, the way our houses are constructed, streets are constructed, how dense we are, to each other, how is our interaction with human beings, or no interaction. That also matters because you need an appropriate level of interaction, human interaction. Or, the trauma and the accidents that happen around the workplace hazards when you're working, the exposure to chemicals, etc. I'm sorry. And so, but equally important is our exposure to pathogens. Pathogens such as bacteria, viruses, fungus, parasites, etc. Prions also. So let me take you through very quickly how our immune system responds to such pathogens day in and day out. Even if you're in lockdown, even if you're in a most hygienic condition, you're not, these days not meeting many people, etc., you still are exposed to a whole range of pathogens, whether you want it or not. And the situation is particularly alarming now with the coronavirus, but even otherwise, we are exposed all the time to the pathogens. And these pathogens like bacteria, fungus, virus, etc., they carry with them what we call as pathogen-associated molecular patterns or danger-associated molecular patterns. These are repetitive glycoprotein motifs. And these repetitive motifs or the patterns are recognized by your innate immune system, the first line of defense, as you must have already heard or read or studied in your high school, that we have what is called first line of defense or the innate immune compartment. You must have heard about tears, the saliva, the cilia, the hair, the mucus, etc., which stops the pathogens entering into your system. In addition to that, the innate immune system also has a whole bunch of cells, the sentinels, the patrols, the police or the housekeeping cells, and molecules, several molecules that are also part of the innate immune compartment. And this innate immune compartment is not specific. It is quick. It is non-specific. It immediately stops the entry of the pathogen, and therefore it doesn't discriminate. It just looks, okay, here is a danger, then I stop it. That's how it works. And that's why those cells that carry receptors for these patterns are important. And those cells, such as antigen presenting cells or the macrophages or phagocytes, they carry what we call as Toll-like receptors. And the Toll-like receptors are pathogen recognizing receptors on the immune cells, on those sentinels, on those phagocytes, etc. As I mentioned, they recognize the pathogen associated molecular patterns and thereby stop them. However, if the pathogen is more virulent and if it's coming in a very big number, and if the innate immune system is not able to cope up and stop the infection right there, then what is called as adaptive immune system kicks in. When the adaptive immune compartment is triggered, it involves essentially, as I told you once again, whole bunch of cells, just like the innate immune system, but much more sophisticated cells. And one of the first cells, which is the link between the innate immune system and the adaptive immune system, is the so-called antigen presenting cell or the dendritic cell, or these are the sentinels, which is shown in the middle there. And these dendritic cells, they carry also the Toll-like receptors, but they carry a whole lot of other receptors. And they catch or they capture the pathogens. They cut them into pieces and process them and present them through the class 1 or class 2 molecule, human leukocyte antigen or HLA, class 1 or class 2 molecules. The presentation of the peptides, small pieces of the bacteria or virus, etc., through the class 1 or class 2 molecules depends on the nature of the pathogen and the type of immune response that you want. So the peptides are presented to T cells through T cell receptors, and the T cells then get activated. The T cells here are represented in blue color. And the T cells then multiply and they become polarized to different subsets of T cells. But they also have another very important function of secreting cytokines. These cytokines, for example, IL-4 or IL-6, they provide signal to the dormant B lymphocyte. The B lymphocyte, which was inactive, dormant, will now become plasma cell and start secreting antibodies. It secretes antibodies against what? Against the little peptide that was shown by the antigen presenting cell through the class 1 or class 2 to the T cells. So that particular piece of antigen is part of the pathogen. And the pathogen, imagine now, has already entered into its target organ, let's say the lungs. I'm showing those different pathogens here in the lungs. So as I told you, these antibodies are specific. Unlike the innate immune compartment, these antibodies are specific. specific to that particular part of the bacteria or the pathogen, which is now lodged in the lungs. So the pathogen is recognized by the antibody. The moment the antibody recognizes through its antigen combining site or the variable portion, the two arms of the Y-shaped antibody molecule, those two arms of the Y-shaped antibody molecule are called the antigen combining site. And when they recognize the pathogen on one side, the constant portion or the single foot-like structure sits on the FC receptors of macrophages, which are now recruited at the site of inflammation or the site of infection. The moment the FC receptor of the macrophages is engaged by the FC portion of the antibody, which is bound to the pathogen, a whole set of biochemical cascade begins, and these macrophages start secreting several obnoxious molecules directed against the pathogen. The idea is now to get rid of the pathogen, and so these are either pro-inflammatory cytokines or other molecules to destroy the pathogen. And if this is not sufficient, the same macrophages also express what is called as complement and complement receptors. And complement is another system of the immune system where a whole range of biochemical entities, they come into picture. And the objective of the activation of the complement system is also to destroy the pathogen by digging a hole or drilling a hole in the pathogen. So if that's not sufficient, the immune system recruits other cells such as neutrophils, which are also able to secrete reactive oxygen species and other obnoxious molecules to get rid of the pathogen. The whole thing is set into motion now. And if even this is not sufficient, as I told you, in addition to complement, if this is not sufficient, then the T lymphocytes get activated and they start polarizing in and start secreting other cytokines. These cytokines are powerful inflammatory mediators. The inflammation here is important. This inflammation is the sign of the reaction of your immune system against the pathogen. We don't have to curb or stop this inflammation. And this will try to get rid of the pathogen. And to do so, the T lymphocytes have a very sophisticated mechanism. They differentiate themselves into different types of T lymphocytes depending upon the nature of the pathogen. And depending upon a very finely orchestrated milieu or the medium of cytokines, we have either Th1 or Th2 or Th17 or Tregs. I'll not go into the details of all this, but in brief, if you have excess of Th1 response, for example, in a bacterial infection, you have Th1. To control excess of Th1, the Th2 cells will come and say, okay, you have done your job, you can stay quiet now. And the Th17 are also very important for intracellular pathogens and some parasites. And they are very powerful cells and they can secrete IL-17 and a whole range of other cytokines. And to control excess of that activity, there's one more bunch of cells called regulatory T cells. These regulatory T cells are very important and they reciprocally regulate the Th17 cells, but the regulatory T cells have a whole range of other functions to control excessive of activation of the immune system. So the net result, what happens here is that the pathogen has to be removed. When you have an intracellular pathogen, for example, a virus which lodges inside the cell, then the T lymphocytes are smart. What they do is they differentiate into one more subpopulation called cytotoxic or CD8 positive T cells. And these cytotoxic T cells, they don't care. They just destroy the entire cell that is hosting the pathogens. This mechanism is also good for cancer because, as you all know, in our body, there is a tendency for the transformation of cells leading to a cancerous cell almost on a regular basis. But this process of immune surveillance ensures that even on a day-to-day basis, if one cell gets transformed and has a tendency to become a cancer cell and starts behaving differently, then the cytotoxic T cell eliminates them, and that ensures the maintenance of homeostasis or well-being. The whole thing works to perfection, and it helps in a very, very well-controlled manner the elimination of the infectious microbe or the pathogen. This works to perfection, as I told you. In addition to that, we have a bonus. Whenever there is an infection, either bacterial or viral, other infection, two different subsets of cells are formed, one from the T cell, one from the B cell. And these subsets are called memory T cells or memory B cells. These cells, as the name indicates, remember what kind of pathogen that was attacked, that attacked. And therefore, they would remember in case the body has to confront the same pathogen once again. They will not allow all this slow process to come in. They will kick in immediately, and they can trigger the immune response very fast to remove the pathogen. The memory B cells do the same function, because the B cells, they stimulate the plasma cells. They stimulate the cells. Is very crucial following an infection. This is very crucial for convalescent plasma therapy. This is very crucial for the very vaccination strategy. And the idea of vaccination, in fact, is to make sure that the body generates these two populations. Because once your body has these populations, memory T cells, B cells, against a whole range of pathogens, then you don't have to worry. The moment the pathogen comes, these two guys kick in and they mount an immune response, and they work efficiently to remove the pathogen. So this is the basic. I know this is not very elaborate, but this gives you an overview of the immune system, immune response, how it happens against a pathogen. But the immune system is quite tricky. It doesn't like any aberration. Any aberration or alteration or deviation from a normalcy can be a problem if there is a decreased immune response, or if it's excessive immune response, or if it is a misdirected immune response. It's not good news. See, this is once again a brief overview of the immune system. And everything was good. It was eliminating the pathogen perfectly. But imagine a situation or situations where a child is born with a defect in any one of these compartments, any one of these components even. And if there is a defect or a lack of any one of these components in your immune system, then you are born as immune deficient. You do not have an appropriate immune response to kill the pathogen. If anything is missing, any one of these missing, and that happens, people have identified so far more than 120 different genetic defects. These are genetic. They have identified more than 120 genetic defects which can affect human beings. And if a child is born with any one of these defects, then that child cannot cope up with any infection, day-to-day infection that you and others, and you and me and others can cope up normally. So there's a bad news. And these children cannot even live outside, because the moment there's a smallest infection, they die because they can't cope up. And these were called bébés bulles in French, or bubble babies, I think, in English. Because without that bubble, which is protected, environment is highly protected, no pathogen, no pathogen whatsoever, the child can grow. The moment the child comes out, it will die. So the image I have shown to you on the right side is that of a child which is immunodeficient, and it is held in hand by Professor Alain Fischer, who for the first time in France, in Paris, not far from where I'm sitting, approximately 15-minute walk from here, he had this ingenious idea of correcting the very gene that was defective and introducing the right gene. And this was the beginning of gene therapy, which happened, I think the beginning happened something like 15, 20 years ago. And a lot of progress has been done. We are not there yet for genetic gene therapy for all these deficiencies, but we are getting there, just for you to know about it. So that was one condition where the immune system, if it is not sufficient, if it is deficient, and this is what happens. But imagine an immune response which is deviated, or an exaggerated immune response. For you are aware of all these allergens that I have displayed on the left side. If you are allergic to any of these substances, then your immune system overreacts. That exaggerated immune response is not good news either. It can range from a very small rhinitis, or what is called common cold, or sneezing and sniffing, and it can be as bad as sloughing off of the skin, or as you can see on the right side. And there are various other forms of allergic reactions which are not good. So the immune system should not be deficient, should not be exaggerated, and it should not be misdirected either. Meaning, imagine the situation where there is no infection. There's no infection, but somehow, for reasons that I'll not go into the details today, the immune system starts reacting. It reacts against what? Against your own pancreas, against your own joints, against your own brain, against your own muscles. The same powerful immune system, which was so efficient in killing the pathogens and destroying the pathogenic cells or the microbial cells, now starts destroying with the same vigor, same violence, your own pancreas, your own joints. All these components come in. And then what happens? If your pancreas is destroyed, you will end up with diabetes. If your joints are destroyed, you will end up with arthritis, etc., etc. And these are misdirected, result of the misdirected immune response, also called autoimmunity. This is not good news either. So what I have told you right now is how a well-behaved immune system works against pathogens, and what happens if it is deficient or exaggerated or misdirected. With this brief background of immune response, let's now try to understand what happens in coronavirus. I'm sure you all have read about it over the last 6 months, but let me take you through this. See, if I had shown you this picture in December, you would have asked, what is this? But today, this particular picture of coronavirus with its spikes and its all flavors, all colors, all forms, all designs, if I show you this picture today and this picture, you will probably ask, who is that on the right side? So much is the popularity of coronavirus. Now, coronaviruses have been around. They are not new to us. They have been around for a long time. Actually, they have been around for a very, very long time. The geneticians have identified, and the population dynamics people have identified, that the coronaviruses have been there for probably centuries and thousands of years. But on and off, they stay in different species. If they stay quietly, let's say in bats or in rodents, they're there without causing any damage to anybody. But unfortunately, they have the habit sometimes of jumping across the species for various reasons that I'm not going into the details today. And when they cross across the species to felines or swines or to camels or to cows, etc., then they cause damage. And we have learned that our famous the type 2, the one which is causing the COVID-19, the SARS-CoV-2, was there in bats, and it jumped across to human beings by crossing over through pangolins or something. There is some amount of data, but then we still need more information to know how it happened. However, we are now facing this huge challenge of the coronavirus infection. And this is the structure of the coronavirus. I'm not a virologist, so I will not go into the very big details of all this, but I'm sure most of you have read and learned so much about the coronavirus, but I don't have to go into the details. So corona is a single-stranded RNA virus. You have RNA as a genetic component, which is inside, shown in blue. And then you have, which is protected by a nucleocapsid protein, and which is then surrounded well by membrane glycoproteins, several layers of membrane glycoproteins, and then the famous spike proteins. So with this brief idea about structure, let us try to understand how it infects the human beings. I'm sure you have already read about it. It can infect through mouth, through nose, or through even eyes. When it hurts or enters through the mouth to the digestive tract, you have quite a few digestive disorders. And if it enters through the respiratory system, it can cause extremely dangerous pathological reaction. Whether it is through eyes, ears, or nose, sorry, not ears, I'm so sorry, nose or mouth, it has to enter into the cells, and those cells are the lining epithelial cells. It has to enter to multiply. And what we have to learn is how they enter into this. We'll take the example of this rectangle, which is, let's say, the lung epithelial cell. Inside is the cytoplasm, and outside the cell, when the virus with its beautiful spikes sits on the lung cells, epithelial cells, it has a preference for a particular type of receptor called angiotensin converting enzyme 2 receptor. It sits on that particular receptor and enters. And when it enters, it uncoats itself into the endosome and then hijacks the entire host cell. I will probably not spend time on describing how exactly the virus undoes itself, uses the host machinery for translation, transcription, and replication, once again, assembly and reorganization, and all the individual components, as I described to you, are remanufactured using the host machinery. I'm sure you have learned about it. And then you have a smooth walled vesicle, and then through a process called exocytosis, it buds out. What you have to know, on the other hand, is if you had had a dinner last night with somebody who is asymptomatic, you did not know that the person had already coronavirus infection, and you picked up the infection this morning, from last night to here, this morning, every cell that was infected with one virus has given rise to 1,000 copies of this virus. But your body is not made of only one cell. It has millions of cells. So millions multiplied by 1,000. This is what happens in one night. This is how strong and how fast coronavirus can multiply. So next morning you have so many cells, which means there is a danger, which means something has to be done. And that's when the immune system says, oh, there is a danger. This thing was not there last week or even yesterday afternoon, and suddenly I'm bombarded with this many unknown new viruses. So I have to do something about it. And that's something you already know. Let's see, once again, very quickly now, I'm not going to as elaborately as earlier. The virus has come in, you know what happens. It is taken by the dendritic cells and it gives a signal to the T cells, and the T cells make cytokines. They give signal to the B cells, and the B cells make antibodies. Antibodies are specific to coronavirus. This takes some time. It takes about 3 to 4 to 5, 6 days. It depends on the quantity of the pathogen you have and the very immune system, how potent it is. Then you also have the T cells that differentiate into cytotoxic T cells, and the cytotoxic T cells, you know what they do. And the macrophages and the neutrophils, they start releasing all these obnoxious cytokines, chemical substances, reactive oxygen reactive species. More importantly, the cytotoxic T cells, through its granzyme and other processes, can destroy the pathogen. The pathogen is destroyed, or the pathogen host cell itself is destroyed. And this works once again perfectly well, and gradually all your coronavirus is eliminated from the body, and you are jumping and kicking in about 5 to 6 days, make it 7 or 1 week. You're fine. Plus, you have the memory T cells. You have the advantage when you have had a coronavirus infection, your T lymphocytes are sensitized, and a little bunch of T lymphocytes called memory T cells, they're lurking in your body, and memory B cells, which means that you had a coronavirus and then you recovered over a matter of 1 week or 10 days or so, sometimes even shorter, 3-4 days. Then you are ready if the coronavirus comes again onto you, because your memory cells are ready. It will kick in and then eliminate the pathogen very quickly. So you're all well. And in fact, about 60 to 70% of the entire population of all of us can come out unscathed without any disease. Meaning you have, like a flu, like a regular flu. You know what a flu is. It's nasty. It's very nasty. You suffer for 3-4 days. All those body ache, headache, muscle, then the running nose, the cough, and all that. Over a period of 3 to 4 days, some people take a little longer, but we come out unscathed. And that's exactly what's happening with coronavirus. Then why this anxiety? Why is this stress? Why is this fear? Why is this entire panic in the world about coronavirus? Why are we going through this frenzy? There are many reasons. The first reason is this is a new virus. As I told you, it jumped across the species very recently. And so our immune system doesn't know it. So it has to fight. It has to make a brand new response against it. And it takes a little time. There's one more problem, which is this virus is particularly extremely contagious. And more importantly, as I told you, this is contagious because we were not knowing this. Our immune system was not used to this, meaning you did not have what is called herd immunity. If if the virus were around for a long time, then we would have been immune to that. And even if it comes again, it would not transmit so fast, so widely everywhere, because people who are already resistant would cut the chain of the transmission. And one more reason why this situation of such anxiety and uncontrolled fear is because some individuals, unfortunately, are not able to cope up with this. These are elderly individuals like me, for example, above 65 or so. And those people who have other underlying diseases like diabetes or obesity or cardiovascular diseases or cancer and many other comorbidities. Unfortunately, there's a whole big population, certain percentage of population that normally live with these conditions. With this, elderly is not a disease, but it's still, your immune system is compromised to some extent. In these individuals, the immune system is not robust, it's not potent, it is not strong, and therefore these people cannot cope up like most young, athletic, dynamic, energetic, healthy, robust people. And in these people, it can cause devastating effects. And that's what you're seeing. And that's exactly what is happening in the world today. So let's see what happens in these individuals. these individuals who have an immunocompromised state because of all those conditions that I just now mentioned, elderly, diabetics, or atherosclerosis, or cardiovascular diseases, or cancer, those patients, when they get the virus, their immune system tries to give the signal to the T cells, and the T cells start making cytotoxic T cells, inflammatory cytokines, and those T cells help the B cells. You know all this. And the cytokines provide signal to B cells. B cells make antibodies. Antibodies are specific, and antibodies engage the macrophages. Macrophages engage FC receptors, and then start secreting the cytokines. The complement also kicks in. Complement causes a lot of damage, and there is more virus coming in, but the immune system is not able to cope up. More virus is coming in. The immune system is not able to handle, but it is trying, it is doing its best. It's releasing the cytokines, all those obnoxious material. And these obnoxious cytokines cannot just be controlled. And the problem is this: when they cannot, there is collateral damage. I mean, they are not only hurting the host, the bacterial cell, they are hurting their own cells. And more virus, more cytokines, more. And that's when this is all proven now. This is proven. The whole profile of the entire range of cytokines in a heat map. This is a very well done study where they have studied close to 40 different cytokines, and most of them are flared up. They are uncontrolled now. And uncontrolled inflammation is not good. I told you in the very beginning that inflammation is good when it is immediately controlled by feedback mechanism. Immune system has that beautiful feedback mechanism, but now it has lost. It is completely in flames. The immune system is on fire in such a way that soon the very immune system which was so useful, so potent against the pathogen, has become against its own enemy now, and it's eclipsed. And this is called the cytokine storm. All those cytokines that I mentioned, they start exerting their effect not just on the pathogen, but on your own coagulation mechanism, on your own lung cells. They cause the cell death. They cause immune paralysis. You have immune depression actually now, and there is septic shock. There is multiple organ failure, and some of us succumb to that. And that's when some of us, we lose them. There is certain percentage of death. That still is not a very, very high number of deaths. Now, what are we going to do about it? How are we going to stop this COVID-19? So what are the different strategies that people are taking? Currently, there is nothing specific, no specific treatment for COVID-19. Whatever we have is from the knowledge of the past coronaviruses, okay? So we are repurposing them and making them. None of the antiviral drugs that is used right now is specific for this particular SARS-CoV-2. So we are only using what was there earlier, but a lot of effort is done to conceive new specific antiviral drugs. I'll tell you a little bit more about immunotherapeutic approaches because that's very interesting, and one or two words about vaccination. Now, coming to antiviral drugs. See, I have simplified this picture now. As I told you, the virus enters, so you can stop the entry. You can stop the entry using specific molecules that are conceived that can block the interaction between the spike protein and the receptor. This can be chemically, designed, or even bioinformaticians like you people can conceive, knowing exactly the structure that is involved in the interaction between the spike protein and the receptor, the ACE2 receptor. And you can conceive molecules chemically designed in such a way, very specifically, that can block. But you have to prove the efficacy, you have to prove the efficiency for how long, and safety, toxicity. That's another ball game. But then antibodies, you can generate antibodies that are specific for either the receptor on one side or any of the spike proteins, or even against the particular region of the combining site. What exactly is the motif? You can conceive antibodies against them and use them. Or there are molecules directed against the membrane fusion and endocytosis process. So chloroquine, which has become very popular, is one such molecule. Or you can target the assembly and the replication of the virus. And a whole range of molecules have already been, well known, well established, because viruses are not new to the pharmaceutical people. They have designed a whole range of molecules from the last 40, 50 years, more than that actually. And you must have all read about remdesivir, how efficient it is. So you have molecules that exist already. Chloroquine became, unfortunately, a little bit of a debate or controversy, because some people said, yes, it is good. Some people said no, particularly a French immunologist from my country, south of France. He was very much in favor of that, but some other people said it is not good. And therefore, I'm not going into the controversy. What we know is it is useful at a particular stage, for sure. One or two words about vaccination. Vaccination is important. As I mentioned, the word herd immunity. You remember, if you all 60% of us, the entire population, get the disease and get over with it and get immune to that, we have our antibodies, and that there is herd immunity. But unfortunately, right now, 60% of the people of the entire population of the world is not affected. It's not easy to get affected like that. Some people even thought about that. Therefore, we need a vaccination. Vaccination is actually the same as getting infection, but without any ill effects. A good vaccine should be safe, it should be efficient, it should evoke the both B and T cell memory compartment, and a single shot should be efficient, and it should have a long-lasting memory. Right now in the world, I think more than 120 different vaccine candidates have been identified, and they are in the process of development. Government agencies, companies, private, academic labs, everyone, 8 of them, or maybe more than 8 now, have also reached clinical trials. But we'll be there. We'll be there in a matter of, let's say, 10 months, 12 months, 18 months, we'll be there. Even in India, quite a few people are onto it. ICMR, CSIR, DST, DBT, all of them are making efforts. Serum Institute of India has entered into collaboration with Oxford University. Bharat Biotech is doing a good job, and Zydus Cadila. Quite a few efforts are made. Now, one or two words about vaccination. So vaccination, I'm sure you all have read about it. You can have a live attenuated vaccine. You can have just a viral vector, take some other virus and use that as a mode of entry into the body. But you use the spike protein. Let's say spike protein has been identified as the most immunogenic of the virus. And therefore, a lot of focus has been emphasized on the spike protein as the antigen, as the vaccine candidate. So use the spike protein, make a recombinant vaccine, meaning you express that, and then you conceive genetically engineered a recombinant spike protein. Or you can use just the DNA that encodes for the spike protein or any other vaccine candidate. Or, in this particular case, even RNA has been very popular. Not an expert on this, but then ultimately the idea is that you need to stimulate your body with a component of the virus that can provoke your immune system in such a way that it makes memory B and T cells, and that confers immunity later on. That's the idea, okay? And all these different trials right now are going on. As of yesterday, at least, or day before yesterday, there were, the virus-based vaccine. There have been less than 10, but that's still there. These are the number of vaccines that are in development, okay? The vector vaccines, replicating or non-replicating, you have quite a few, up to 25. Then the DNA-based or RNA-based vaccine, they're also there up to 20. And the protein-based, the one which is most common, either protein subunit or virus-like particle, they're the highest. And then you also have other vaccines like the BCG, polio. There are a lot of very interesting studies, because BCG was initially, as you may remember, once again designed here, right here, Institut Pasteur Institute. long back against, probably this is the most widely used vaccine ever. Every year, more than 1 million kids are injected with this vaccine. So this vaccine is supposed to protect against Mycobacterium tuberculosis, but then we also know the cross protection is quite important. And there are some interesting studies based on BCG, even in coronavirus, actually. So what should a vaccine do? Vaccine should induce, just like earlier, it should be taken up by the macrophages and then presented to T cell, B cells, as I told you earlier. And you have the cytotoxic T cells will remove that. And the same thing, any vaccine should do this, okay? It will neutralize the virus. It should neutralize the virus. Or it should provoke, actually, all this. Because there is no, this is the very vaccine is the infection now. So you're provoking the immune system. But most important is that it provokes the generation of the memory B and T cell, okay? And these memory T and B cells will be sitting there, and if there's a reinfection, they'll get into action and remove the pathogen. Let's hope for a vaccine soon. I'll spend a few minutes now, last few minutes, on immunotherapeutic and other approaches. Well, as you all know, the patients undergo a very serious lung dysfunction and therefore respiratory dysfunction. They need oxygen therapy and mechanical ventilation. When there is an excessive of inflammation, at one stage, as I told you, there is an excessive inflammation, you don't need an immune stimulation. On the contrary, you need immune suppression, and that's what corticosteroids do. BCG, I already mentioned to you, it provokes a kind of a large spectrum immune boost. And traditional medicine, India, we have Ayush. There's a lot of effort made on traditional medicine, but then I'll spend 1 or 2 minutes on immunotherapy. Talking about immunotherapy, there's a whole range of effort made on immunotherapy. See, if you remember, at one point I insisted on cytokine storm and how important are cytokines in terms of provoking inflammation. But this inflammation, as I told you, is important. But when it is out of control, you have to stop that. Therefore, there are approaches using monoclonal antibodies directed against IL-6 or IL-6 receptors or IL-6 antagonists or anti-TNF alpha. There are quite a few cytokines that are targeted using most commonly the humanized monoclonal antibodies or chemical substances that can neutralize the cytokines. That's one effort. Secondly, the NK cells. NK cells are important in killing the host cell that hosts or that lodges the receptor. So the virus, I'm so sorry. So there are people who are trying to conceive NK cells from the hematopoietic stem cells or purify NK cells, immortalize them, and see whether you can use those cells as cytotoxic cells, actually. And then you have regulatory T cells. As I told you, regulatory T cells are probably one of the most important components of your immune system, which can control an unwanted excessive T cell activation or many other functions. We in the lab have studied quite a bit regulatory T cells, and there are approaches. This is still slightly, let's say, infant stage, this particular approach, but it can come in handy if we know exactly at what stage you can use, and if you have the right regulatory T cells, it can control that excessive activation of the immune system. Then we'll discuss a little bit about convalescent plasma and immunomodulation using immunoglobulins. They are mostly related. In the convalescent plasma or hyperimmunoglobulin or neutralizing monoclonal antibodies or in immunoglobulin therapy, we are all talking about the same thing, ultimately the same thing. That is the antibodies or immunoglobulins. whether it is convalescent plasma or hyperimmune globulin, hyperimmune serum, or neutralizing monoclonal antibody, or IVIG. I'll mention that in a few minutes. We are all talking about one single molecule at the end, ultimately. That is antibody. And to tell you a little bit about antibodies, I have to take you back to 120 years ago. How did this all begin? What is plasma therapy? What is antibody therapy? How did it start? It all started way back, about 120, 130 years ago, when a certain Emil Adolf von Behring with Kita Sato identified for the first time that if you take a toxin or a bacteria, let's say in case of their work, it was Clostridium diphtheria, and its toxin, they went on injecting guinea pigs in a small dose. Over a period of time, the guinea pig's plasma had some substance which was able to neutralize the toxin. This was one of the probably the most significant discoveries in terms of treatment of infectious diseases. The moment they understood that there is a substance in the serum or plasma, they injected animals that were infected intentionally, and they showed that your plasma that came from animals that were hyperimmune, meaning you had boosted that, was able to neutralize and protect the sick animals. This was a major discovery. And soon they identified that in this serum or plasma, you have a substance called antibodies. So for me, this work is probably one of the most important in terms of biotechnology, for which von Behring got the first ever Nobel in medicine in I think it was a little later, of course. I don't remember the age, the year exactly. As soon as in 1892 or so, when this discovery was made and shown that you can use the plasma to treat passively an infection, Émile Roux in France, not far from here again, he treated 300 children that were going to die of diphtheria. All that he did was purify, meaning he did the same thing as Emil von Behring, made huge amounts of plasma against the toxin, and they used anti-diphtheria serum or plasma. You know the difference between serum and plasma, of course. The blood minus the cells is plasma. Okay, and this work was done not too far from here, Pasteur Institute. So the antibodies that were thus discovered by Emil von Behring became the central preoccupation of immunologists for decades and decades. And even today, we have not understood exactly or completely how this works. Functional aspects were studied. Structure and binding properties of these antibodies were studied. More importantly, the therapeutic aspects, how to exploit these antibodies.
Découvert qu'on peut isoler les anticorps diphtérie.
So then, I'm sorry about this. So many antibodies were discovered, and Spanish flu in 1918. Okay, sorry, there was a crosstalk. It must have been a slide that I picked up. So what is important is that as soon as the discovery of antibodies was made, many pathogens were identified at that time, like diphtheria, tetanus, botulinum, anthrax, and many. Okay, so what they did was these smart doctors, Debré, Nicole, Cornsey, they purified the pathogen and injected, and either the toxin or the pathogen, and they had in their drawers antibodies which were ready now. Same thing was done in 1918, the famous Spanish flu that we are talking about, but there were mixed results those days. So now one or two words about convalescent plasma therapy. Convalescent means somebody who has recovered. Okay. In the case of all these, we were injecting the animals. They were, let's hope they were recovering, and they were collecting the plasma from those animals that were recovering. Now, the principle for convalescent plasma therapy in coronavirus is many people are recovering. As you know, as I told you, 60% of us will recover. I told you, we have many people around us who have recovered. So you just have to register yourself. Go there, give your plasma. And this plasma is the source for treatment. But you need a large quantity, unfortunately, half a liter of plasma that is injected. Right now, 60 clinical trials are underway. This plasma therapy, as I told you, it's not new. We know about it. We know a lot about it. But unfortunately, we know also the disadvantages or the demerits or the concerns, because sometimes this plasma therapy may even worsen the infection, because actually this is called antibody dependent enhancement. An antibody recognizes this and then it provokes the immune response, or it will in fact worsen the infection. And this has been shown in some cases actually. And the preformed antibodies may block the active immune response. Therefore, in children less than 2 months, for example, you don't give a vaccination, because it may, the mother that has transferred maternal antibodies that have been transferred are quite important colostrum antibodies. They protect the child less than 2 months against many infections. But at that time, if you give any vaccination, it will be not efficient, because these maternally transformed antibodies can block. So sometimes this passive immunotherapy is not good. And elderly individuals may not tolerate that huge amount of plasma. And you may have some allergic reactions. And it's also shown that in people with lung damage have difficulty in breathing. And more importantly, the safety, because you are injecting the whole plasma. And although right now we know how to screen against a large number of pathogens, you never know what is there. Sometimes if you miss any of this, there have been disasters in the past. Transmission of HIV, transmission of hepatitis, transmission of prions has happened because of plasma therapy. So one should be careful. Now, let me go back to this. So when these people identified, something else happened. Soon after that, World War II began. Okay, and the World War II was not good news for many people, right? Not just for the soldiers dying at the battlefront. Many soldiers were dying because of infections, and there was no antibiotic at that time. They were dying because of infections or body fluid losses. And at that time in Boston, Edwin Kahn had identified how to separate the plasma into all its components, or at least most of the components. And antibodies is one of the very important components. And so is plasma albumin. And even today, when you order albumin, you will see that it's called Kahn's fraction 5. This is fraction 5 of Kahn's precipitation. It's an alcohol precipitation. The reason I'm mentioning this is because this was the beginning of what is called antibody therapy. And when he discovered antibodies, this was a pool of immunoglobulins. I'm going to cut the story short here, but what I would like you to know is that there is an antibody preparation called IVIG, also called intravenous immunoglobulin. Intravenous immunoglobulin because it is injected by intravenous route. It's actually a pool of antibodies from several thousand healthy people. The fact that several thousand healthy people are pooled, it is conceivable, at least theoretically, that they're exposed to a whole range of pathogens, and therefore they would have antibodies against a whole range of pathogens, such as including coronaviruses or cross-reactive antibodies. Or you have antibodies to some self-antigens and antibodies to antibodies. I'll not go into the details, but if you have any questions, I'll be happy to answer them. And this IVIG, in the beginning, it was used in the treatment. It's always used in the treatment of immune deficiencies. You remember at one point I told you that there are some children that can be born without immune system fully functional or some defects. The only way they can survive at this stage is through IVIG or bone marrow transplantation. And there are attempts of using IVIG in COVID-19. So on one hand, you have monoclonal antibodies that can be highly specific. There are some that are being conceived that can be used. Monoclonal, I'm talking about monoclonal humanized. Or these are polyclonal antibodies, or you have plasma therapy. I hope you have understood these 3 different ways or 4 different ways of understanding the antibodies. One last slide before I leave you. So, in my opinion, we are on the road to recovery. More than 1 million people around the world have already recovered, and mathematical models estimate that 99.5% of the people will recover. There will be more new and specific antiviral drugs. Vaccine trials will begin soon, and I'm sure coronavirus will be controlled and can be controlled and will be controlled. I thank my people once again, all my colleagues, and I'll not name all of them, but I know that they know that I'm thankful to them. I thank you, merci beaucoup, and I'm all yours. Ritika?
Yes, sir. Thank you, sir, for a great talk. So we will go ahead and take some time for the questions now. The audience can still post their questions in the comment section if they haven't done that already. So meanwhile, we'll collect all the questions from our live chat. I'm going to ask some questions that we received during the registration of this webinar. So the first question, sir, is does getting infected by one strain of coronavirus give a potential immunity against another strain?
Yeah, in principle, yes, because when we say one strain of corona, I'm not a big virologist. What happens is, you remember at one point I said, in your body overnight you have 1,000 copies that are done. When 1,000 copies are done, it's a human after all, and there may be a small defect or a small change, and it's not 100% foolproof. So there will be some, differences in these 1,000 copies. So far, over 700 different such small differences have already been identified. So if the question is, if you are reinfected with a virus which is slightly different, whether your first infection will confer immunity, answer is mostly yes.
Okay, so another question we have is, what could be the immunological responses against the vaccine if we have one for the virus?
Good. Immunological response against the vaccine is the same as the immune response to the virus itself. If we are talking about when we say immunological response, it is the same as the one against the virus, which I showed you in one of the last slides. Probably when the question was posted, it was before that and before. So you have either it is a viral-based or vector-based or DNA or RNA vaccine. It will provoke the same kind of immune response. But what is most important is you have a memory T cell and memory B cell compartment. Right.
Okay, so how does a person get affected by the virus in spite of being healthy or fit and having a strong immune system?
You get affected because you have not seen this virus in the past. When you say affected, we are affected by many pathogens day in and day out. And depending upon the load of the pathogen, the virulence of the pathogen, you mount more or less, relatively corresponding immune response. But you normally get over with this. If your immune system is competent enough, you get over with this and you will be recovering without any major adverse effect. Some of them will lose. In the case of coronavirus, we are talking about losing your smell and some other side effects. Unfortunately, some of them have had neurological or digestive consequences. But in general, healthy, that's what is the question. If you are healthy and fit, you normally get over with the immune response.
All right, sir. So we have another question which says, why do the reports or warnings or guidelines from WHO keep on changing, even though we know a lot about the virus already?
I'm probably not competent enough to answer this because I'm not WHO. But WHO is essentially guided by scientists, and the scientists keep on gathering the information. And science is such a beautiful domain that you can never be sure. One day, what you say today may not be sure because more discovery will be done tomorrow. And so, obviously, if you have to change the recommendations, alterations, you have to change. You have to adjust accordingly. I think they do for the benefit of the people, I guess.
Okay, sir. Um, we have another question. I think one more question from the registration. How is immunotherapy more beneficial over plasma and EDTA chelation therapy in finding a cure for SARS-CoV-2?
Okay, as I mentioned, immunotherapy has has different approaches or different targets, and if one is uh, able to identify the window in which the patient is going through, your immunotherapy can be extremely potent and extremely specific and get over. And plasma, as I mentioned, it has only component that is beneficial is the antibodies. It's also efficient, but it all depends on when you approach the patient. Depending on that, immunotherapy will be more or less beneficial, and one cannot say one is better than the other. No, it is one. It depends on the patient, depends on the approach, but they are all more or less equally efficient.
Okay, so now I'm going to move on to the questions that we've received on our live chat. So the question is, if a patient is already suffering from autoimmune mediated interstitial lung fibrosis and is already on steroids and suffers from high blood pressure, what is the possibility of COVID-19 prognosis in such a patient?
Yes, this is a good question, but unfortunately, as I mentioned to you, these are those unfortunate small number of patients who have the comorbidities. In these patients, the consequences can be serious because already if they have on steroids, meaning they're immune compromised, obviously they're in a very bad shape. So one has to be much more careful. They have to be entered immediately into care, intensive care, and they have to get oxygen therapy plus all the other approaches. But yes, the question is apt. If these are the people who are going to suffer most, and some of us will lose them. Yes.
Okay. Another question is, are there vaccines designed for earlier SARS outbreaks? If so, can they be modified and made effective against for this pandemic?
Answer is yes, because as I told you, this is called repurposing, and we are trying earlier SARS, although it's not exactly identical, it until we come out with a real vaccine specific, which is there are many trials. I told you there are 120 different candidates, and 8 trials are already underway. And yes, until that happens, there is nothing wrong in we can try to use earlier vaccines, and there are some attempts made on that also. Yes.
Could we expect the coronavirus situation to become more like flu once we have successfully come up with a vaccine?
Yes, I'm quite optimistic. Although today, India, Brazil, and some countries, even the United States, we are going through certain, unfortunate peaks and surge upwards. Soon, there will be appropriate vaccines, there will be treatment, and we will get over this, I hope, in a matter of few months. Although, some mathematical models say the peak has not come in India yet, but I'm sure we will be successful and get over this, particularly the vaccine herd immunity that will establish soon.
Okay, so I think we don't have any more questions here. Then I think yeah. So here we wrap up our Q&A section. I hope all your doubts are cleared. Thank you for participating in this discussion. For any queries or to get any regular updates from us, follow us on our social media handles or write to us at covws dot in at gmail.com. Also, please find a link to the feedback form for this webinar in the comments section. It is a great resource to really get individualized feedback from you. As a follow-up to this webinar series, we have an upcoming webinar on 6th of August 2020. The talk is titled as The Role of Academic Institutions in the Current Pandemic and will be delivered by Dr. Rakesh Mishra, Director at CSR CCMB Hyderabad. We believe that this particular webinar is a great platform to understand the importance of academic advising during the pandemic. So we hope you'd join us. From there, though, this concludes our session for today. Have a great day, everyone, and thank you again, sir, for presenting this. I really think you put together everything very nicely, and it was really easy for even people with no formal training in biology to understand what happens once we encounter the virus in our body. We really appreciate your effort and your expertise. Uh, Please stay with us for some more time, sir. And yeah, I'd like to wrap up the webinar here for the audience. Thank you very much for joining us today, and we hope to see you next time.